It's my pleasure to introduce our next guest from Forge Biologics, co-founder, president, and chief executive officer, Dr. Tim Miller, and chief technology officer, Dr. David Dismuke. As a reminder to attendees, if you'd like to ask a question of our speakers, please type it into the question box under the video player. Tim, David, really happy to have you joining us today. And to start, Tim, could you provide a couple minutes of introductory comments about Forge for investors who are new to the story?
Yeah, sure. First, thanks for having us on today. David and I are really looking forward to some great commentary and questions. And it's been an exciting year for Forge as we're all coming out of this pandemic and starting to see more and more conferences come up live and getting to share the story out a bit more. Currently, Forge is about to hit its 200th hire an employee. So, the brief timeline that you'll see a little bit is we closed a Series A in mid-2020, a Series B in mid-2021. So, jumping certainly through a lot of hoops of growth in this past time frame has really been a remarkable state. And as we're finishing the last of our build of our 20 GMP suites, having a number of them already online and up to 1,000-liter scale. We're going to have a number of announcements coming out in May and June, which we can't wait to share, both on some of the offerings that we have and just, really just talking about some of the additional progress in the company. And, really one of the ways that we communicate through for a lot of our colleagues, for investors, for clients and business development as well, is really through some of the videos to be able to show a lot of our story. You can hear a lot about David and I talking, but sometimes a picture or a video tends to speak a little bit more clearly about some of the progress. So we're going to show something for about maybe I think it's about 10 minutes, and then we'll come back and I think we have a couple of questions and going through a Q&A session. So, Kate, we're good to go if you're ready to roll it.
Yeah, that'd be great.
Welcome to yesterday. The familiar monotony of waiting in line at a CDMO. We know the feeling. It's why we exist. To solve for the pain points we ourselves experienced when building a gene therapy program. We had a chance to envision what a better tomorrow could look like in this industry. We envisioned, we assembled, and then we got. Tomorrow has been decades in the making. With an unparalleled team of experienced experts, combined with the right facility and equipment, Forge can deliver the highest quality AAV products in the world. And we're doing it at a scale the world has never seen before, using bioreactors ranging in capacity from 1 liter to an astonishing 5,000 liters. We are taking the expectations of what's possible in this industry and transforming them. This is where your therapy comes to life. This is where hope for patients becomes real. This is the new standard for excellence in the world of AAV manufacturing. This is the future. And the future is Forge.
Forge was founded on the belief that with the right people, you can push the limits of what can be accomplished in a gene therapy company. With the team we've assembled here at Forge, we're breaking down barriers in the gene therapy manufacturing space.
Our mission is to enable access to life-changing gene therapies. And to do this, we've adopted a unique business model as a hybrid CDMO and therapeutics company. As a contract development manufacturing organization, we offer end-to-end AAV manufacturing, from research grade through to cGMP production. As a therapeutics company, we have a pipeline of clinical stage AAV-based therapies. The synergies between our CDMO and therapeutic programs enable us to improve our platform process faster and gain insight as to how we can serve our external clients better.
Building hope is not something that just happens. It's not passive. It requires intention, dedication, and commitment. and guides us to be better every day. When we develop or manufacture therapies for rare genetic diseases, we're driven by a deep desire to build hope.
Scientific curiosity, when paired with an exceptional team and diverse scientific experiences, is a recipe for innovation. This approach means we dedicate ourselves to solving complex technical problems, approaching obstacles with an open mind, which often leads to new and exciting scientific advancements.
When we remove the bottleneck in gene therapy manufacturing, therapies will come to life at a rate the world has never seen before. We realized early on that when you solve the manufacturing problem, you literally save lives.
Every element and detail within our build is intentional, and our facility was designed to uphold the highest levels of safety, quality, and precision. And the fact that it's a beautiful space, that's just a bonus.
The team we have here at Forge is taking on the world of gene therapy to innovate, to inspire, and lead the way in accelerating a brighter future for patients and their families.
Forge Biologics is a new kind of gene therapy CDMO. For our team of industry veterans, providing novel therapeutics is about enabling access for patients safer, faster, and at scale. We are gene therapy developers, researchers, and clinicians, and we built Forge Biologics to solve for the pain points we ourselves experience when moving programs through development and into the clinic. We are here to provide our collective expertise, technology, and services so that your therapies can get to patients who need them the most. We see ourselves as partners in this endeavor to create a lasting impact, and we look forward to bringing your mission to life.
Our analytical development team sets the standard against which your products will be tested, and we set our bar high. Using a standard battery of platform assays enables an integrated and efficient development pathway. Our service is unique in that we offer in-house analytics, development, and qualification, saving our clients time and indirect fees associated with outsourced testing. These services include analytical ultracentrifugation, next-generation sequencing, capsid identity by mass spectrometry, and product-specific replication-competent AAV.
Our full suite of manufacturing services were built to support our clients at any phase of development. We offer services from small-scale optimization studies to 50-liter scale research-grade productions, toxicology and engineering runs from 50 to 500 liters, and cGMP manufacturing from 50 to 5,000 liters. The 5,000-liter bioreactor has a unique 10-to-1 turndown ratio that allows optimal flexibility and scales. Our facility is specifically designed to optimize throughput. The upstream and downstream processes occur in separate rooms so that we can turn over suites faster.
At Forge, we know time is the enemy for our clients. That is why we built a platform approach that sets your gene therapy program up for safety, scalability, and speed. Our technology includes our HEC 293 suspension ignition cell line and P-AMBA ad helper plasmid. To ensure safety, our P-AMBA and helper plasmid has an improved safety profile engineered to reduce known immunogenic elements. For scalability, our ignition cells are a clonal cell line with clear, well-documented history that are optimized for scalable suspension growth. Additionally, our platform approach reduces timelines by employing Forge's proprietary technology and in-house support services to streamline process development and GMP readiness.
We have developed a platform AAV manufacturing process to streamline our clients' journey from discovery to the clinic. By understanding and optimizing upstream and downstream process parameters, we can confidently produce many commonly used AAV serotypes with limited product-specific development. We are continuously advancing our knowledge and expertise to refine our process parameters to generate AAV. Leveraging the platform process, the Forge ignition cell line, and high-throughput development tools like the Ambr 250, we work with our clients to quickly advance their therapeutic programs with productive, robust, and scalable production of AAV with improved purity and safety profiles.
Many gene therapy companies and universities in the research development phase need access to research-grade AAV to quickly identify lead therapeutic candidates. Manufacturing research-grade AAV marks the beginning of large-scale and commercial production of your vector at Forge. Our research-scale AAV offering is quick and allows you to get to know our team and technology before moving on to further development.
We've made quality a priority by building it into our foundation. We have implemented a cloud-based quality management system that supports our mission of enabling access to life-changing gene therapies by reducing regulatory compliance risk, improving our staff efficiency, and delivering timely and accurate quality metrics. This provides assurance to our clients and patients that they will receive the highest quality products from our team.
Navigating the regulatory landscape is burdensome to many gene therapy developers. It's a catalyst for why we offer regulatory support services to our clients. Forge staffs an in-house regulatory team that stays up to date on the most current regulatory guidance so your team can focus on developing your therapy. We provide support from preclinical through commercial development, including assistance with responses to agencies' requests for information, review of the client's CMC or IMPD information, and program regulatory strategy.
Our dedicated client team is devoted to ensuring your program is taken care of every step of the way. From understanding our offerings to technical advisory, our team is here to set your program up for success. We work with clients through initial introductory calls, technical deep dives, on-site visits, contracts detailing your scope of work, and project kickoffs.
Our fundamental focus on patients is at the heart of everything we do. We are ready to collaborate with your team in accelerating access to life-changing gene therapies to help reach those who need them the most.
Well, that's a great video. Really glad that you were able to show that and share that with everybody. Let me just dive right into some of the Q&A. And I think last year at this conference, the overwhelming consensus was that there was insufficient supply to meet demands for development and manufacturing services, and this was creating scheduling backlogs. So from your perspective as a CDMO, what's the current state of CGT manufacturing capacity?
Yeah, it's an interesting still perspective on the landscape, and I think that many of us saw a lot of announcements in 2020 and 2021 of companies putting a lot of money to work to try to either expand, current manufacturing capacities or even do a greenfield build out. And look, the market has been challenging. We all know that. I think that what we see is that many of those companies have pulled back on their manufacturing plans. And rather than put, 80 or $100 million into a hole in the ground, it's take 10 or 15 million and go and find a CDMO and try to get your manufacturing in order to get to the next value inflection point. So, I think that that's been, what we've seen in the current, landscape in cell and gene therapy. And, it's also a reflection that building out a workforce like this, the talent, the workforce is really difficult to come by. And, even when you get some of that, it takes a really long time to develop your cell lines, your quality system, and everything around the build out. So, I think, there are a number of great of our colleagues that are doing a great job trying to continue to build out their manufacturing capabilities. And, we'll continue to see how the field, really absorbs that additional capacity that's coming online. But again, from our perspective, we're still seeing a significant shortage in supply for good quality manufacturers. David, I don't know if you want to add any comments to that either.
Yeah, I would just say we definitely have not seen anything slow down. It's it may be difficult to completely assess because as we've grown, I think we've gotten more income inbounds, clients coming in, but we haven't seen any slowdown in new clients coming into Forge.
Kind of a newer wrinkle since the start of the year has just been the valuations in the market. And so recently we've seen a number of pre-commercial stage biotech companies announce strategic initiatives designed to extend their cash runways. And that includes headcount reductions, but also pipeline rationalization. So, what are you seeing in maybe the last couple of months that is either translating into increased demand for your CDMO services or maybe an increase in rescheduling requests?
Yeah, I think have seen an uptick since the end of last year in the number of clients requesting proposals and clients that are, really trying to drive forward. They're rather to put money to work with experts in the space. And so, we see this translated in an increased demand for CDMO services. There's always going to be good funding for good science, and we continue to see, more programs, more AAV capsids continue to get funded. So, from that perspective, again, good science is often independent of market, but you still see, market pressure putting how much work can actually get done. So I think, one of the old expressions, right, is, good science always gets funded. Just you're hoping that there's more funding to continue. And look, we anticipate, like everybody else, that the market will turn again soon. So I think that that's kind of where we're seeing, that inflection point in the space.
Yeah, and I think we've been able to capitalize on it, if anything. One from maybe how people are spending money, but also, as you pointed out, with a headcount and as we're growing, to be able to bring in talent that maybe had to leave somewhere else.
Yeah, interesting. Want to dig deeper into some of your proprietary AAV design components. Can you talk about how your proprietary helper plasmid and RepCap plasmids are helping to enhance gene expression and improve safety?
Sure, I'll take this one there, Tim. So that, we really tried to take a very thorough approach when we built Forge to make sure that we're giving the best offerings possible instead of using what was, on the shelf, or been commonly used for years. We took those materials, whether it's the cell line or whether it was the RepCap or the helper plasmids, and tried to engineer it a quality by design approach to make sure that we were, again, giving the best offering. Specifically around our ad helper plasmid, we've created dozens and dozens of different versions of that ad helper to really optimize productivity as well as the safety feature, notably, removing viral elements that are not necessary while still maintaining high yields. And the same thing for our RepCaps. We've been able to design RepCap plasmids that give 10x improvement in product quality or product yield from some of the capsids. So, those are the aspects that really, I think, help our clients. And then for our cell line, we have a very thorough. I'm confident that we have the most thorough HEK history that's available for the cell line, and that the productivity side, we're really trying to reduce the amount of impurities, specifically around empty full. So typically we're around 40 to 50% of full particles at the time of early production without an enrichment step. And so that makes our downstream processing much easier to be able to get to those high, 90+% full particles, again, reducing the amount of impurities as much as possible.
So, just kind of following on that on the QC side and your analytics, what are you doing to improve the performance and reliability of your assays to characterize potency, purity, and other important product attributes?
Yeah, I think that's it's a fundamental piece of what you need to do as an AAV CDMO. And I think, again, probably one of the strengths of Forge is that we have, a long history in working with these assays, whether they're, a ddPCR for your titer or for residual impurities, for example, empty full, as we mentioned earlier, testing that through analytical ultracentrifugation. We've built our own RCAV lab, in a segregated part of the facility to be able to test and a faster means for our clients to ensure that there's no replication competent AAV in their product. And then specifically, I think we have a poster coming up at American Society of Gene and Cell Therapy in May. We'll be presenting on some of the improvements that we've made around our residual host cell DNA assay. I think it's been a difficult thing for people in the field to test, and we've made a number of improvements to that assay to get better specificity and better sensitivity in the assay, so we can provide that to our clients. So again, I think just overall taking a very deep approach to how we're providing analytics for our clients.
Well, great. I mean, and clearly it's an important area, especially as we think about the clinical holds we see. And I guess maybe from your perspective, how much of this is related to sponsors advancing poorly qualified products into the clinic, or possibly due to, regulatory agencies having learned on the fly of what things are more important to look at? Is it goalposts moving, or is it just goalposts coming clearer into focus as products get deeper into the clinic and closer towards commercialization?
Yeah, it's probably a little bit of both. It's not 2008 anymore, where 95% of phase 1 gene therapy trials failed in development. So, as the agency, I think, has gotten a better, more and more trials have come on, they've had to place additional scrutiny on both the manufacturing and really what's considered efficacy too. So manufacturing has always been a key component of that. It's only come under increased scrutiny over the past 3 or 4 years. So, I think it's a little bit of both. Agencies trying to move along with sponsors and provide guidance, as much as techniques and purification strategies, for example, or upstream continues to advance. So I think in the future, I think that things will become a little bit more set. I mean, ddPCR, for example, is now really considered the standard rather than qPCR. So we'll see more of those things kind of, I think, settle out soon.
Okay, well, let me squeeze in one last question here, and it's kind of gives you a chance to summarize everything we've just reviewed here. But how is Forge differentiating itself as the preferred AAV CDMO partner?
Yeah, a couple different ways. As David mentioned, our cell line is one that you can take from research grade through clinical to commercial. The full traceability of it, the fact that it produces higher fulls, right out of the gate. These are all things that, many developers aren't able to provide to their clients. We'd also probably say that Forge differentiates on the number of release testing that we use, the ability to use our own adenoviral helper plasmid, and really scale. You'll see, we've talked a little bit about our 5,000 liter bioreactors coming online soon, and we'll put out a couple more announcements around that around probably ASGCT. But so being able to go up to that, those are some of the biggest, if not the biggest, HEK 293 suspension cultures in the world to be able to do AAV production. So a number of different ways to do it. If you look at a case study around Duchenne, you really understand just how much liters need to be produced just to treat like 25% of the prevalent population, and that's just one indication.
All right, well, Tim, David, thank you so much for joining us today.
Thank you so much.